Hello. Today I'd like to introduce you to a new algorithm for data generation and augmentation that we call Sugar. Sugar offers a new paradigm for data synthesis that models intrinsic geometry of data rather than density. Traditional generative models infer a distribution that estimates data density and resample it. We note that such generation can distort the shape of the data and lose sparse regions in rare examples that are often of interest in data exploration. All of these factors bias the output of contemporary generative models. In contrast, Sugar learns a manifold geometry from data and then augments it to uniformly resample this manifold. Let's illustrate this process with a simple example of variably sampled points from a manifold that we've embedded in R3. To learn a data manifold, we'll use random walks over local neighborhoods. These define a nonlinear diffusion geometry for the entire data. Next, Sugar generates new points along this manifold. Sparse regions are filled in by synthetic points obtained via careful perturbation of the input data. Then, we combine the synthesized points with the input data. To do this, we use a recently proposed measure-based kernel that is robust to variations in sampling density. This allows us to form a new diffusion geometry, which we use to uniformly resample the data, which, in this example, corrects sampling biases along the shoulders of the plane. In our paper, we demonstrate several examples, and here I'll briefly cover two of them. For our first example, we show that Sugar can improve imbalance classification by augmenting undersampled class manifolds and outperforms other methods. Next, we use Sugar for exploratory data analysis. A major intended application for Sugar is in high-dimensional biology experiments, where rare cell populations are of great experimental interest. We applied Sugar to a single-cell RNA sequencing dataset consisting of 4,000 cells and 13,000 genes. Here, Sugar effectively captures the cellular state space and as a result is able to synthesize hypothetical cells from a critically undersampled transition of developing B cells. In the paper, we are excited to confirm via genetic analysis that this trajectory is realistic and follows canonical biology. The uses of Sugar are beyond just what we've outlined here. It is a new paradigm for data generation, which we detail in our paper in the 2018 NeurIPS proceedings. Thanks for watching. We can't wait to see you at NeurIPS 2018 in Montreal.